Hi, Mr. Worski here today, and this is the third screencast of our DNA series. This is going to be a short one. We're going to talk about mutations briefly here. And we know that mutations are random, inheritable changes in DNA. So if the mutation is passed on to a body cell, that can cause cancer. But if it's in your gamete, it can be passed on to the next generation, and you can pass those mutations on to your offspring. So the different types. The first type is mismatch or base substitution. So on the left-hand side, we have the normal DNA. On the right-hand side, we have the mutation. Right? So the mutation would be here, which is the C instead of the T. It looks like I did that wrong and should have an extra T in there too. Oh well. So the possible causes of a mismatch is DNA polymerase making a mistake. The insertion is when we add one or more DNA bases, like we added this A here, or we add this T here, and the possible causes of an insertion, because you're actually inserting the DNA, is unequal exchange of DNA and crossing over. You might be asking yourself, what? Well, we're going to talk about crossing over in a little while, a couple of different modules down, or a couple of different units down, and you'll know, but just keep that in mind. A deletion is when we remove one or more DNA bases. So we have two T's here, we only have one T. We have two T's here, we only have one T. So that's possibly a thymine dimer caused by ultraviolet light. Hey, we just talked about that. I know. So it looks like we've deleted T's from the DNA bases. It can also be caused by unequal exchange of DNA during crossing over. You think, what? Well, we'll get to that momentarily, soon. Inversion is when we flip a section of DNA. So this section from the T's right there, from the A's here, where the T's there is only taken out and flipped around, right? And that can occur during crossing over. Man, this crossing over sounds like a real drag. No, it isn't. It provides us a lot of variation, which is good. Fifth, breakage or loss, we just basically cut off the DNA, and that can be caused by a bulky lesion. Right? So it stops DNA polymerase from going down the DNA. You don't have DNA synthesized. It can also be caused during crossing over when the piece of DNA just floats away, which would be a drag. Extra copies, when we have extra copies of DNA, and we have that with the CCTT. CCTT is copied over and over again. And a possible cause of that is unequal exchange of DNA during crossing over. DNA crossing over again. I know. So those are the 6 different types of mutations. And we are done with the screencast for this evening. So hopefully you got all the information in your notes. If you have any questions, please feel free to ask tomorrow in class, or read about it in your book, or watch the video over again. Heck, it's only like 3 minutes long. Have a good night.